Okay, welcome back everybody to the IMAG MSM Working Group on Multiscale Models in Viral Pandemics. The June 22nd mini seminar. We will have two speakers sharing the slot today, Yob and Harm. As always, I need to remind you that this meeting is live streamed and recorded and archivally available in public. Tom and I are always available to take your suggestions and complaints, comments. Jim and Lorenzo are here to help out and do a lot of the lifting as well. We have our communication channels. We have the GLIMPRINT.org website. Please help us populate it. Tom has done an amazing job creating that website. Please help us make it into a real thing. We have our usual Twitter handle, MSM Viral. Maybe we can get a little bit more exposure if we use that hashtag. Next week, I will be seeing a lot of people, I hope, if we're not all sick, at Bethesda for the NIH 20th anniversary meeting of IMAG MSM. Our group is one of the big successes. The fact that we're still meeting with 170 talks is a big success for them. I think it's too late for people to register, but I know a lot of people will be there in Bethesda next week. Look forward to seeing you. We still have the open faculty positions in in medical digital twins here at IU. If people are interested, contact me and let me know. Tom has been working hard on GLIMPRINT. John Rice made a comment that we need to define what that organization is supposed to do and do it. We are going to have a meeting tomorrow to discuss that. Please help us out. At the moment, we're advertising the fact that we have our CompuCell workshop first 2 weeks of August this year. We have 170 people registered for it so far. So it'll be interesting to see how that goes. We have an IMAG steering committee meeting tomorrow at 11 a.m. If you need to be there and don't have the invite, please let us know. Again, we need to be serious about thinking about what we need to do going forward after almost 3 years working together. Are there any other announcements before we continue? We have a number of seminars scheduled. July 6th, we have Tom Yankeloff from University of Texas. We're doing incredibly detailed digital twins for cancer therapy. A very impressive program combining computer modeling, machine learning, clinical deployments, therapy design, and basic wet lab biology. July 12th, we are going to have Anna and Reinhardt and perhaps others summarizing the results of the Paris Immune Digital Twin Workshop. We have a July 20th meeting scheduled. Going forward, please help us fill the roster. Let us know what you'd like. Suggest yourselves, your students, your postdocs, the people you'd like to hear from. We definitely want to get an agenda that responds to people's interests and needs. Today we have Yob and Harm talking to us about Towards a Virtual Embryo, computational modeling of neural tube closure defects.
Well, thank you. It's good to see that there's so many people online. Thank you for the invitation. We're presenting our work here, and like James said, we're gonna talk about the computational toxicology of neural tube defects. I will be giving an introduction in the topic, and like more on the biology behind everything, and then Job will will give a talk on the technical details of the model that we aim to build, or that we actually are building at the moment. My name is Harm Heusinkveld. I'm from the Center for Health Protection at the National Institute of Public Health and the Environment in the Netherlands. And the project we're running all this work under is the ONTOX project, which is a project aiming at a 3R approach for repeated dose toxicity studies. So replacing animal studies with alternative strategies. Hopefully then we can all have them free of rodent studies. But one of the things that we're looking into is in silico modeling and agent-based modeling models to in the end predict toxicity of compounds. Within the project, there are 3 topics focused on liver, kidney, and developmental neurotoxicity, and we're in the developmental neurotoxicity part with our neural tube closure. And the thing about this project that makes this project stand out from most other projects is that everything we do and all things we develop for these 3R approaches are rooted in human biology. And the way we do that is by the build of physiological maps. And based on the physiological maps, so we're building ontologies, which is actually a description of or a combined description of physiology, together with adverse outcome pathways, describing everything that goes wrong from the first contact of a chemical to the adverse outcome, in our case, the neural tube closure. And based on this, we're building the testing strategy, but everything rooted in human biology. And in other projects, but that's a side note, we're taking the same approach for Parkinson's disease and non-genotoxic carcinogenicity to be able to mimic what happens in the human. So let me start out with physiological maps and why do we do that? And the phrase I like is to model something, you have to understand it first. And that holds in particular for strategies for toxicity testing. And let me show you an analogy. This is a map of Paris, and imagine that I'm traveling through Europe and I have to switch trains in Paris. Then I have to let me see, is there a pointer somewhere? There's supposed to be a pointer. Well, in the upper right corner, you can see the Gare du Nord station, and all trains from the Netherlands end there. And it's a hard end. The trains are only going back and not going forward anymore. And if I want to go to the south of France, I have to move to the lower left corner of Paris, Gare Montparnasse, to switch trains to, for instance, Bordeaux. But if this is the map I have, then I won't go anywhere, because depending on the map, and if this is the map, I'm going in the wrong direction, may end up in dead-end streets because I have no idea where they are. And at least, even if I have a couple of hours, I will never be in time for the train going south. So, in fact, better maps are needed. You can see my cursor. Oh, that's great. Then I will just move it on purpose. So instead of this map, we need a better map of Paris to be able to catch this train. And in fact, this is more what we would love to have, which is a map within the background, a lot of detail, with exact points where we are, Gare du Nord, and how we come to Gare Montparnasse. With different options of going there, starting from the same point and going to the same point, but having different routes. So this is basically what we would need to travel Paris. And the same holds for the ontologies. To be able to predict what happens going from one point to another, we need a map. And thinking of human physiology and maps, then you can, I always use this map, which is the quote unquote map of life. And if I want to predict what chemicals do to this map, that you might end up with an adverse outcome, being a disease or a neural tube closure defect, then I need to know where my tipping points are, where my points of no return, where is this roundabout, where is this street corner where I need to switch directions, and then I can draw my lines and my route, how I come from chemical interaction with the physiological system to an adverse outcome or a disease. And I'm going to answer questions later if you don't mind. Sorry. And if I have this map and I have drawn out all the key elements and the turning points and all the stop signs, then based on these key elements or key events, if we're moving forward to adverse outcome terminology, then I can build a testing strategy based upon which I can predict what are the odds of going from the left side of the map to the right side, or from the chemical interaction to the adverse outcome. And with this test battery that I can devise based on the key events and some kinetics, which are on the left and giving input there, if all this information feeds into a virtual human in silico model, or in our case an agent-based model, then I can come up with a risk profile of a chemical, and I can estimate what the risk is of exposure of a certain compound. Okay, well, in our case, it's neural tube closure. A little bit on the background why this case study was chosen. Neural tube closure is a process that forms the basis for the spinal cord and the nervous system in the developing embryo. It's something that happens really early in pregnancy, so at the end of third week, by the time you are maybe or not even aware that you're pregnant. And we know from literature and all toxicity data that this process can be a target for a chemical disturbance that may result in neural tube closure defects. To give you an idea, Spina Bifida, which is the most commonly occurring neural tube closure defect, you will see it in 3.5 out of 10,000 babies born in the US. It's a debilitating disease. These children that are born, the ones that are actually born alive, will be severely handicapped. And we have at the moment no test available for risk assessment, so we are not able to test whether a chemical compound is able to disturb this process and may cause spina bifida. So we need a new test strategy, or we need a test strategy, basically. We set our goals at charting the physiology underlying neural tube closure, starting with a manual literature search with one linking pin, and that's one of the important proteins in the whole process of neural tube closure, which is retinoic acid. And at first we looked at all available model species, and in our case that was human, mouse, xenopus, zebrafish, and chick. And we listed all the relevant processes and the most relevant processes, and try to build an overview of what was happening. And here you can see like a sort of more or less artist impression of what happens within the middle of the schematic overview of the closing neural tube, or the early embryo. And on the left, 6 stages, left and right, 6 stages of neural tube closure. I'll come to that in more detail in a minute. And these lists that we started out with were lists of genes and proteins and cells involved in spina bifida, genes, proteins, and cells involved in the pathway from revolving around retinoic acid. And at the same time, identifying most important cell types and functions and factors that were influencing these genes and proteins. And to come to finally a definition of all the critical steps in neural tube closure. And this meant determining key phenotypes, keeping an eye on that. There are several drawings of what it looks like out there in literature, such as the one with the 4 pictures you can see here. But we kept at all time a view on what is happening in real life. So the EM picture on the right, where you can see, okay, well, this is what it should look like. This is what we're aiming for this process. And all this information we use to build a computational model of this process. And when I say we're building lists, long lists. One of the examples is this one. You're not supposed to be able to read this, but this is a list of literature with cell types, behavior of cell types, and the signaling pathways that play a role in this. And then we started to draw these maps based on these list maps in Cytoscape. And these ones escalated pretty quickly to something that Italian people would call spaghetti, but we found it unedible. So we said, okay, we need to do something else. We need to make something that is more insightful. And one of the first things we did is looking into the species, what are species differences in this case. And we said, okay, well, there is human data available. We're aiming ultimately for the human system. This data is available, but it's fragmented. And so we need something to complement that. And we realized quickly that mouse data was abundantly available, and the species concordance between mouse and human is pretty good. So we were able to use mouse data plus human data and build our physiological map out of that. We set a time frame for ourselves. So we said, okay, well, we're going from flat neural plate to closed tube, and that's it. The moment there is the EMT and the migration of the neural crest cells, so they're migrating away, and when once the neural tube is closed, we said, okay, that's the end of what we're aiming for. And with the decision was made to indeed take the retinoic acid as a linking pin. There are a lot of other processes going on, but we said, okay, retinoic acid will be our pivot point here. And we realized that we're missing some pathways in this case. And one of the most important ones that we're missing is one of the rescue pathways that many doctors and physicians are aware of, because folic acid is we know that it's able to prevent neural tube closure defects. And we know how it works, but this pathway is not in our physiological map yet, because it's based on HDAC inhibition and has only sideways something to do with retinoic acid. So we have built a physiological map based on retinoic acid, and we're aware that it's not complete yet, but actually it covers most aspects of neural tube closure pretty effectively. So in the end, we came to a bit of storytelling, a bit more advanced than the one I showed you in the earlier pictures, showing where the different protein gradients are that are important. So the BMP gradient coming from the surface ectoderm, the sonic hedgehog coming from the notochord, retinoic acid, which is based in the paraxial mesoderm, and we pictured this way our starting point and our end point. And instead of using Cytoscape, we moved on to CellDesigner. And this is a really, really neat platform to build physiological maps. It's all in systems biology markup language, or SBML, and you're able to really draw the processes that play a role. From gene expression to protein phosphorylation and etc. And what you can see here is our physiological map that we ended up with, and the different boxes represent the different cell systems and all the processes that are relevant in these systems. And these are this is a lower number than the processes that are actually all going on at the same time, but we reduced our processes to the ones that are absolutely necessary for neural tube closure. And if processes were not necessary for neural tube closure, or there was a knockout data available saying, okay, well, you can knock out this gene, but neural tube will still close perfectly fine. And in most cases, we left them out just to keep it as simple as possible and as complicated as necessary. So within the ONTOX project, we have a collaboration with the University of Liège in Belgium, where there's a group of Lisbeth Geres, and they're working on building physiological maps. They're not doing anything else, basically. and really good at it. So they helped us building the 2.0 version of our physiological map. And in the end, we moved on from CellDesigner, which is SBML based, but still limited in what you can do with it, to the Minerva platform, which is an online tool. And the fun thing about it is that it's editable. So if the guys in Liège post our physiological map online, we are able to annotate or edit the map in real time online too. And that's one of the things that is fun about Minerva is that it's basically built in several layers. You have a top level, an activity flow, and a process description. And you can build these layers really to navigate the maps. And here you can see an example of this modularization and the structure of the maps. This is an example of the liver case study from ONTOX, where you can see in the top level, which is really the top level, it's the organ and the cell system. And from there, you can navigate to a much more detailed map. And within this detailed map, you can even navigate to really parts of physiology that are worked out in even more detail, and those ones are interoperable with other online tools such as wiki pathways. And the fact that it's online and it's publicly available really speeds up development of these maps, because in the end everyone can add knowledge or put question marks at where you think it's going wrong. So really on the top level, in a little bit more detail here, this one is about liver lipid metabolism, or in terms of adverse outcome, the pathway towards steatosis. And what you can see here is that you can see this liver and the liver cell in the liver cell, and on the surface of the liver cell, the processes that are involved in steatosis, or liver lipid metabolism leading to steatosis. Again, just as simple as possible and as complex as necessary. And from here, from all the different processes that are outlined, you can click into the more detailed processes underneath, really giving you an insight. So for the neural tube, we're following the same lines, but to build a computational model that Job will tell you about in a minute. This one is rooted in also in human biology, even though there's a lot of mouse biology in there. We know from biology and physiology that the mouse data that we have in there really is homologous to the human. And based on this physiological map, we can build a testing strategy with several test systems in it to really be able to capture the risk of chemical exposure for neural tube defects. And models that we're thinking about for this testing strategy are complex cell models based on stem cells, for instance, for rosette formation, which is very It's a really nice in vitro self-organizing system where the same biology is as in the neural tube in vivo, but just part of the process. And at the same time, we're also thinking about whole organism in vitro models such as zebrafish embryos. And earlier I said that we skipped the zebrafish data. And that's basically because zebrafish, the neural tube closure in zebrafish is phenotypically different from the human or the mammalian system. But on the level of gene regulation and protein regulation and signaling pathways, it's more or less the same. So we're investigating the options for having a whole organism model where we can generate transcriptomics or genomics data with to feed in our computational model. And in the end, we hope that our model will be suitable for next generation probabilistic risk assessment, because if we feed our genomics fingerprint of chemical exposure to our computational model, it's hopefully it will give us the probability of neural tube closure defects if you run it for 10,000 or even 100,000 times. So with that, I think I'm going to move the floor to Job, who will tell you more about the agent-based modeling and how he built the model for the neural tube closure.
My name is Berkhout. I'm the PhD student of HARM, affiliated with the RIVM, Dutch National Institute for Public Health and Environment, as well as Utrecht University. And on behalf of the ONTOX project, I am currently working on this computational model. I'll first introduce the model, go over some aspects of the model, and then show what you can do with it once you incorporate enough biology in this model. So, towards an ontology of neural tube closure defects and very broad steps, and Harm just showed you the physiology and the determination of key phenotypes. And I will going over the building of the computational model. And in the near future, we're also going to perform a model validation study, of course. So computational modeling of neural tube closure. We use the great platform, open source platform, Computational 3D. And Computational 3D is a multicellular agent-based model, which means that cells are represented as individual agents or objects on which sort of behaviors can be attached. And here in the image, you see we use the cellular plots model. You see the cells on the left-hand side. On the right side, you see sort of the agent-based model, and you see the cells divided in sort of like squares, small pieces or pixels or voxels, you can call it. And these voxels, they can move around. They, based on the behavior, what you give to them, more like a sort of a video game, you could say, they make decisions. This cell that's moving around doesn't do much, probably not that many behavior attached to it. But once you make a more advanced model, you can do quite some with it. And here you see our neural tube closure model built in Computational 3D. And as Harm showed with neural tube closure, we have neural fold elevation, the parts sides migrate towards each other. We see formation of cellular structures in there. We see some sort of bending of the tissue, and the sides fuse together, and we have full neural tube closure. So what is in this model and what can we do with it? Well, I'll first talk a bit about spatial organization. We based on human physiology, different protein gradients that guide certain behavior patterns, which we mined from the literature as Harm showed you earlier. And also one important aspect is cell-wise formation. So the spatial organization of the computational model. Here you see the computational model again here in the middle. And if we then compare it to actually a human model, the first human embryo, which is an open source platform where they mined human embryonic data, you see here the embryo, sections of the embryo we're modeling in 2 dimensions. And there are quite some similarities between our computational model and a human embryo section, actually. You see here at the top right, you see the neural fold elevation. The neural folds go up with the neuroectoderm layer here, middle, just as the embryo sort of goes upright. We see some of equiconstriction here on the sides, bending of the tissue, same in our model, and also summation of large cellular structures which are surrounded by extracellular matrix. Again, quite some similarities between our model and for the human model. But as Harm mentioned, we use mouse data as well to be able to achieve this. And if you compare to a mouse, you see again a lot of similarities. That is, of course, because the human neural tube closure and the mouse neural tube closure has a lot of similarities as well. Again, you see here neural fold elevation, bending and closure of the neuroectoderm, and then you have the full neural tube formed. So to go into the next component, equiconstriction, which is a very major driver for proper neural tube closure. And for people who are not aware of this concept, equiconstriction is basically sort of creating bottle or wedge-shaped cells. You can see here in this image, see here that the basal layer becomes thinner, the layer becomes broader and you see sort of a bending of tissue, bottle-shaped cells, apical constriction. And if you place it then into sort of a system as neural tube closure, here on the sides, you see the dorsal to hinge point formation. Again, the bottle-shaped cells, very critical for neural tube closure. So how does apical constriction work in the neural tube closure? And apical constriction is mediated by BMP and sonic hedgehog, bone morphogenic protein and sonic hedgehog. And what we know from literature is that BMP induces apical constriction and sonic hedgehog inhibits it. And what we found in knockout studies is that BMP goes too high, it's inhibited the apical constriction. So probably if you have an apical constriction on a signaling level, the requirement or the probability of apical constriction to occur is influenced by BMP in sort of a hill-shaped function, and it's more like a downwards function for sonic hedgehog. And by calculating the probability of apical constriction to occur, and these are not actual graphs, this is just for illustration, of course. By calculating probability, we can sort of do a probability assessment, or what's the probability for apical constriction to occur under the influence of these signals. So that's what we use in the model. So based on these protein signals, the cells decide whether or not they will perform this sort of behavior, this apical constriction. And then how I program it into the model, or to create bottle-shaped cells, I attached multiple cells towards each other. I sort of made springs to approximate the ectomycin machinery of the cell to keep them together. And then you reduce the size of one volume, increase the size of the other volume. And then you see sort of this kind of bottle-shaped cell structure that we see in biology as well. It is a very important driver for neural tube closure. Then I will shortly introduce somatogenesis, which is also an important part of our computational model, and then show what we can do with this computational model of ours. So somat formation is basically a very important part of neural tube closure. They exert also a lot of physical forces, but helps with the bending. You see somatogenesis in chicks, and you see that cells sort of self-organize into these round structures. You see in this image, they were sort of hanging out in the matrix, and then they self-organize in these round shape-like structures. So how do we do this in our computational model? So what we basically see is we have extracellular matrix in between our cells, and you see the cells being in there, their shapes similar to what we see in biology, and then we differentiate the cells to a somat cell which has different properties and behaviors that makes it self-organize and clump together. That happens in the somites. And if we compare the shapes of our mesodermal cells compared to biology, see a lot of similarities. We see that sort of the wiggly shapes of the cells that's represented in our model as well. And of course, if you then look at the somites, the structures, it's actually completely different. We took the approach to create somites in a much more simplified structure. Actually, somatogenesis is very complex. There's actually a somatogenesis model out there as well, in which James also took an important role, and Julio Belmonte. And we decided to go to approximate the physical forces that somites create during neural tube closure and get the more simplified structure, still exerting the physical forces necessary to go from a flat ectoderm towards a closed neural tube. So if we have a neural tube closure model with different mechanisms, then what can we do with such a neural tube closure model? Well, the idea of these computational models is that it's probabilistic. You can ask, so to say, what if to the model, and the model will give you a scenario, what could happen. So let us ask the question, well, what would happen if we change something on level of BMP signaling, a very important driver on apical constriction? So here we have a normal situation. So what if BMP signaling is normal? And then we see, again, neural volt elevation and closure of neural tube. But what would happen if we upregulate BMP? Well, we see actually a very similar pattern as in the normal situation. See that the bending maybe starts a bit earlier on a different location, and the model looks a bit more stiff, but in the end, it managed to close as well. And we just see a very nice neural tube closure. Then if we add more severe perturbations, what if we hyperactivate BMP, reduce BMP signaling by 75%? We see that there's like no those lots of information away on the top of the neural tube, and she still migrates towards the other. See that the sides or the top surface ectoderm is going to close, but the rest of the neural tube actually is not able to make it. And this is a phenomenon we also see in vivo. When BMP is hyperactivated, we see a very similar pattern in mouse studies, where really the top of the neural ectoderm is severely bended, and the neural tube actually, it does close, but only the surface ectoderm. And we could call this phenomenon, we could call this Spina Bifida, or closed Spina Bifida, for example. And then on the other side, what if we hyperactivate BMP? Well, what we see is that there's actually barely any bending at all compared to the other situations. See that it starts with some bending, but in the end, the model becomes really stiff and completely open. And again, this is also a phenomenon we see in vivo studies, where with the hyperactivated BMP, there's no dorsal transport formation, and the closure is severely disturbed. So we also have another example. So what we can do with this model, say we disrupt formation of the floor plate. And the floor plate is basically this part of the computational model. It secretes also an important protein gradient called sonic hedgehog. And what we see in a disrupted floor plate formation is there's sort of like a cellular clump on the bottom of the neural tube. And again, this is very similar as what we see in vivo studies in mice. Very similar adverse outcome, so to say. However, if you recall the morphologicatory signals of constriction, sonic hedgehog, which is secreted by the floor plate, inhibits those transport formation. So because this signal is removed or severely reduced, cells actually show much more apoconstriction compared to the normal situation. So whilst their floor plate is removed and the cells form sort of a clump because of an induced and increased amount of apoconstriction, the neural tube still manages to close. And this is also what is seen in vivo studies. So even without a floor plate, the neural tube is able to close. And we are able to recapitulate this in our computational model. So to conclude this short demonstration of our work, we showed the first steps towards a biologically relevant computational model of human neural tube closure. And we also managed to demonstrate adverse outcomes similar to in vivo studies. Of course, we're far from finished. There's only a part of biology in there. And the map that Harm showed earlier is actually quite vast. We need to add much more biology to there. We're going to add much more biology there in the future. And to further improve our model, we want to do case studies with chemicals from different classes to see, all right, what would a chemical perturbation do to a neural tube, and how can we simulate it and predict it with our computational model. So, of course, I did not do this all on my own. My great promotion team, including Harm, Aldert, and Juliette, are helping me, as well as external advisors on the level of computational modeling, as well as developmental biology, with Tom Knudsen, Richard Spencer, James Glazier, and Julio Belmonte. And of course, the ONTOX team gave me the opportunity to do this work in there. So this is the end of my presentation. If you want to know more, you can contact me. So the RIVM spam filter is very strict. So if I do not respond, you can try my personal email address. And if there are any questions, I'm happy to answer them.
Okay, great. Time for questions. Lorenzo, go ahead.
Yeah.
I don't have a great connection, so it's better. I think I spell out in the chat. So in general, my question is about how did you choose the rules and you map the arrows that you have in both simulation to known kinetic laws. How did you choose that? How do you parameterize that and so on and so forth? The usual question, I'm sorry.
Yeah, very good question, Lorenzo. At first, we did it in a qualitative way. We know the directionality of the arrows is similar in human and mice. We will move on to a more quantified network in the end, and this is one of the things where we're digging for data, where we're trying to find the data underneath. So I hope that answered your question a little bit, but you got a question for Job too. So,
I mean, how do you decide whether a heel is better than mass action or I don't know, Michaelis-Menten or any other kind of functional form? That is the hard one because we never know. But
yeah, that's true.
Yes, that's an excellent question. Well, as Harm mentioned, we first focus on qualitative data. So how I approach it right now is because we don't know the quantitative aspects of all the reactions. So we take the same simplified Hill equation for our probabilistic assessments of the protein gradients, and based on model optimization studies. So actually with chemicals and actually more generated data, we hope to optimize those functions to have a better decision into whether to use a Hill function, a Michaelis-Menten function, or a mass action function. But for now, we use a simplified approach, use scaled Hill functions for all our behavior patterns. And at a later stage, we're going to improve them based on measurable data.
thank you. That all makes a lot of sense to me.
Okay, great.
Tom, go ahead, please.
Yeah, thank you. Very beautiful presentation as always, and it's great to work with you guys. So one of the things that we've liked about CC3D models is their stochasticity, and they don't give you the same outcome every time you run them. And I'm wondering, have you done a dose response to look for probabilistic outcomes? So if you say, take your BMP model, if you run them 20 times over different levels of perturbation, can you make a determination that, say, percentage of neural tube defects is associated with X amount of BMP alteration?
So it's definitely possible. I haven't done it in the depth that you suggested, but it's certainly something we want to do once we have additional biology into the model. So for now, I ran the simulation multiple times, but not in a very tight dose response, but much broader steps as I showed in the presentation. And next step will be a really tight dose response estimation, and then also a probabilistic chance for a neural tube defect to occur.
For now, we took the sort of all or nothing approach. And we were happily surprised that it worked. It worked out really nicely. But that will indeed be the way we're going to move forward to the probabilistic to do the probabilistic assessment of the effect of chemicals, making a much tighter dose response curve. So yeah, of course.
And if I could ask another question. So as you know, in humans, the spina bifida is primarily a lumbar occurrence. And as you also know, segmentation of the neural tube is regionally specific. Do you have any sort of thoughts? I know we talked about this in the past, but talks about, say, reconstructing models across different segments of the neural tube and reconstructing, say, a three-dimensional system?
Yes, I haven't showed my latest version of the model currently, but I'm currently working on a updated version where I can create segments along the axis of the neural tube. And first start with that to be able to approximate three-dimensional modeling as best as possible. And depending on those outcomes, we can consider whether we want to move towards an actual three-dimensional model. But of course, that adds a lot of different complexity and uncertainties to it as well. So we need to make that decision once we tested the different segments across the axis of the neural tube model. It's a great suggestion.
Yeah, one of the things I would like to add to that is I would love to have this sort of reflected in the physiological map as well. If there is a biological reason for the fact that neural tube closure defects always or mostly occur in the lumbar region, then this should somehow be captured in the physiological map or in the description of biology. But that's, yeah.
Yeah, and that's, of course, one thing where humans and mice differ, because in mice, it's the opposite. They're more prominent anteriorly than posteriorly. Yeah.
Exactly. That's why we need to make a very detailed species comparison up to the level of detailed as possible. Because, yeah, absolutely.
Other questions? And I have a question which I've talked about with them before, but which I'll ask here, which is, how do you quantify the outcomes? You have these beautiful movies, but how do you quantify what the different phenotype is of the output of your simulation?
So we talked about this before. So I have a number of metrics. Not all of them are implemented in the model, but the most promising metric we like to use is to be able to do something on the level of machine learning, where we can be able to quantify the shape of the neural ectoderm. Ideally, there's quite some data out there. For example, I showed the virtual human embryo images. If we can properly segment those images, we can actually do a quantitative comparison between actual biological data of our model and what's out there in the literature. And that is one of the ambitious goals we want to have. What we want to try to achieve, to be actually be able to compare it to in vivo adverse outcomes, or computational simulations. And other more low-level metrics are, for example, the distance between cells, to be able to see if it's closed, whether the left side or the right side is touching, whether the left side or the right side of the surface ectoderm is touching, to be able to differentiate between closed and open spina bifida, as well as the apical constriction probability of the cells, how many cells go in apical constriction, other cell counting approaches. But machine learning approach would be very interesting and very promising to use in this situation.
I think for publication purposes, if you did some, say, image J imaging of your samples across multiple runs, and look for real sensitive changes, I think that's going to really make it much more palatable in terms of publishing these models. I would certainly encourage you to, think about image J analysis.
Yeah, image J would be a very great option to use for this, as well as maybe deep learning approaches, which I developed in the past on a low level and with only a black and white situation, may be very well approachable to actually compare it, of course, in combination with MHA to segment the neural ectoderm from the rest of the cell layers. So
yeah, I like the idea of image analysis. I think that would indeed make it more understandable for people how you approach this and how you actually quantify the outcome.
Yeah, that's a great suggestion.
I could keep asking questions, James. I don't want to.
Go ahead. No. Listen, I mean, people know the format. People can speak up and ask any questions they want.
I wanted to go to something you said early on, Harm, about the rosette formation being a way of quantitating some of the cellular changes. Have you thought about working with Randy Ashton a little bit on that?
We did. I've been talking to him at SOT about the neural rosette, his neural rosette model, and we're currently looking into the availability of different models, and then Randy's model is one of the candidates. We have another protocol that we stumbled upon in I think it was University of Constance in Germany, where they built the same, more similar, comparable model neural rosettes, and we're considering comparing Randy's model with that model and seeing if with the different differentiation protocols and the different approaches, whether you can come to the same answer or whether they will give you different answers. But Randy's model really looks funky.
So they just got our ToxCast plates, and they're going to start testing well over 1,000 chemicals for region-specific effects on human neural rosette formation that simulates neural tube segmentation. So that's really, I think, a very good connection for you down the road, because your models will translate what his in vitro data shows.
Exactly. Yeah. That sounds like a gold mine of data for our model. Yeah, absolutely.
Thank you.
Yup, on the bucket list.
Yeah. Yes. I write it down.